Hello, my name is Grace Murray. I'm going to be presenting my biolab presentation, and I am going to share my screen with you. Okay. First, start out with one of the figures that's in my presentation, how I made it. So we can see here that this is the figure that is in my presentation with the antibody titer test for each individual, and they did this 3 separate times, and we can also see the standard deviation error bars, and I'm going to show you guys how I got this. So you can see here that I have the same values. I have the standard deviation for each individual. Their average samples, just for my own doing, it was easier for me, and standard deviations of each of the samples. So I'm going to make the graph, same graph that I have in the other one. Okay, so once I have this graph, you can see it's the same as I just showed, and then we're going to add these error bars, doing this by each series and adding a custom specified value and adding in these standard deviations that I calculated through Excel. Do this for each of them. Oops. Second series, and then the third. So you can see I'm doing all of these in their positive error value bars because all of my standard deviations I calculated were all positive, none of them are negative. So, okay, so you can see that this is the same as I got here. These are how I got my standard deviation bars, and now I'm going to be presenting my presentation. Is this it? Okay. Okay, so like I said in the beginning, I'm going to be doing this presentation for my final biolab exam, and it's on SARS-CoV testing and how we do it and all that kind of stuff. So an introduction to what SARS-CoV-2 actually is, it's a virus strain of SARS, which we know is a severe acute respiratory syndrome, and this originates back in China in about 2003, and it's actually from a civet cat, which is more of like a mongoose type animal in our country. And the SARS-CoV-2 is the actual virus strain that causes the disease COVID-19, and so that's what causing the actual virus strain of the SARS is what's causing COVID-19, and it's genetically similar to the SARS virus, but it's a whole different virus, a whole different strain. And what the coronavirus is actually a term describing an illness that causes problems with your respiratory system, which we know makes sense because a lot of the symptoms people who have it have been having a lot of bad respiratory symptoms, and it originates from an infected animal who spreads it to a human, spreading it to other humans, which is called a zoonotic spillover event, and we'll talk more about that. But the current coronavirus outbreak is called COVID-19 and originated in Wuhan, China in December of 2019, hence the COVID-19. So a little bit about viruses. A virus can be defined as a microscopic parasite that infects living organisms, specifically in our cells, which are the only place the viruses can replicate. So we know that the virus pretty much goes to our cells and attacks it and gets inside of our cells and starts replicating itself in the cells, kind of just making the cells structure shut down. A virus can usually contain either RNA or DNA genetic material, and the SARS-CoV-2 specifically infects RNA of patients. So this classifies it as an RNA virus versus a DNA one. It is a positive sense in a shingle strand, which we know is an RNA virus, and it belongs to the coronavirus family of viruses. So how did this virus spread so aggressively? We know there's been many theories about how the virus started, how it spread, and whatnot. So the most popular was that it originated in bats in Wuhan, China, and it spread to humans and eventually causes worldwide pandemic. So that goes back to the zoonotic spillover event I talked about, that a virus originates in an animal, which then spreads to a human, and each of the humans spread to each other, causing whole world to be infected. So this virus is spread specifically by respiratory droplets such as sneezing and coughing. Spread these water droplets that can contain the virus, which is, pretty normal when we're sick, when we sneeze and cough. Respiratory droplets can be everywhere, even when we're just talking, these droplets can come out. And so these droplets also can contaminate objects. So if a person who had the coronavirus coughed or sneezed on a table, and their respiratory droplets can contaminate this table, and anyone can touch this table, contaminate a table, and then maybe touch their mouth, touch your eye, which can infect themselves with the virus. And it also can be spread by aerosol transmission, which are the particles that contain the virus that are so small they can stay in the air for hours and travel by air currents, which is pretty terrifying to me. So currently in the United States, we have more cases than anywhere else in the world, with over 3 million confirmed cases. In my home state of New Jersey, we have one of the highest confirmed cases in state in America at about 130,000 almost, causing us to be in a statewide lockdown. So I am in pretty much like the epicenter of the whole virus in the country. So how do we test for this virus? So there are many ways we can test for this virus. One is a PCR testing, and a PCR test is a polymerase chain reaction. And what this test can do is it can copy one specific part of your DNA and massively multiply it to be analyzed to diagnose a disease, or in this case, detect an RNA sequence of SARS-CoV-2. So basically, we know how small DNA sequences can be. So what this test does is takes a specific one and just multiplies it to trillions and millions of them so we can test them for a disease rather than just having one little one. So this test is made up of a mixture of target DNA, nucleotides, an enzyme called Taq DNA polymerase, and DNA primers. So basically, the first thing we do is we denature the two strands of DNA, getting rid of its hydrogen bonds by boiling the DNA strand at 95 degrees Celsius, which makes hydrogen bonds go away. So we just have the two strands. And then we lower the temperature to about 45 to 65 degrees Celsius to anneal and have the DNA primers that we know are in the mixture now bind to the separate DNA strands and kind of take place for the hydrogen bonds that were previously boiled off. And the third step of this PCR test is known as extension. The temperature is raised for the third time to 72 degrees, so the enzyme Taq DNA polymerase can add nucleotides to the end of the already attached DNA primers and multiply. So basically what this enzyme does is the enzyme is the part that multiplies it. It's the third step, and that's what the PCR cycle is, these three steps of denaturing, annealing, and extension. And we know that one cycle contains these three steps, and one cycle doubles every single time. So the first cycle, we have 2 DNA strands. The second cycle, we have 4. Third, we have 8, and so on and so on. And we need to do about 40 full of these cycles to actually be able to analyze the target DNA, which is crazy because that's about a million billion DNA strands when we actually start out with only one. So unfortunately, this PCR test would not be applicable to test for SARS-CoV-2 because it's an RNA-infected virus, and PCR specifically focuses on DNA strands. So what we do use is something called an RT-PCR test. It only accounts for, PCR only accounts for DNA, and since SARS-CoV-2 is an RNA virus, we must find a different strategy of this particular test, the PCR, and we do a different strategy called RT-PCR, which is real-time polymerase chain reaction. And so to perform RT-PCR, we take a nasopharyngeal swab, which we have seen people getting tested for them. It's a really long swab. We need to stick it really far up to be able to get as much DNA as possible because we know that we can't detect it, just one little one. So we know that there is not enough viral DNA to detect directly in the patient's swab, so we use RT-PCR to amplify many copies of this viral RNA segment of the specific N gene using 2' and 2 probe sets. So these primers bind to the target RNA N gene, which is similar to the DNA primers that binded to the DNA strands after the hydrogen bonds were boiled off. And so the enzyme, which is different in RT-PCR, is reverse transcriptase, and it extends the RNA primer the primer DNA of RNA being tested, synthesizing complementary DNA or cDNA. So that's basically what's starting to amplify each of it and synthesizing our complementary DNA. So after that step, it's pretty much the same as what we just talked about in PCR. So that Taq DNA polymerase enzyme comes in again and extends that second strand to produce a double-strand copy of the SARS-CoV gene for us to amplify and test after going through many cycles. So we want to amplify that gene to see if something binds to it, to be able to see if this person contained SARS-CoV RNA. During amplification, we add a fluorescent probe, which I talked about in the beginning, to go to a specific target sequence between the primers to try and detect the RNA gene to see if this test is positive or negative. So during the extension phase, that same enzyme again, Taq polymerase, actually breaks down the probe that's binded, which causes the dyes to separate from each other, makes a fluorescent signal. So when we see this fluorescent signal, we can see that these primers and the RNA gene were detected to be binded, and they make a neon little line from the fluorescent probe, and that's how we can detect that SARS-CoV in this test, and we know this person has tested positive for SARS-CoV-2. So another test we can do, this one is more for someone who thinks they had the virus in the past. This tests our antibodies, which we know fight against our immune diseases, and we can see if we had a certain disease by testing a person's antibodies. So this ELISA test stands for an enzyme-linked immunosorbent assay, and they're used to test a person's antibodies from a sample of their blood to see if they had a certain virus by measuring the concentration between the antibodies and antigen interactions. And what we know about the antibodies and antigen interactions is the antigen is pretty much a receptor protein on its cell surface, and then it binds to specific foreign molecules, and the antibody binds to its specific antigen, creating an immune response to foreign molecules, which we know is what happens when we get sick or have a virus or disease in our body. So there are multiple types to carry out this type of ELISA testing. We will be focusing mostly on the indirect way, and that which requires two antibodies. The first one binds to its correct antigen, which is placed, and the second antibody is enzyme linked. So we carry out this testing by adding the antigens of SARS-CoV-2 to a plate, like a microliter little plate, and we add the patient's serum. And what a patient's serum is, it's from their blood, but it's kind of like a toned down version to get out like our red blood cells, anything like that, just to focus mostly on their antibodies. And we want to see if their antibodies that from their serum are binding to the specific antigen that's already on the plate. So after they bind, or if they do bind, we add the second antibody, which has an enzyme attached to it. And what this enzyme does is it can change the color of a substrate that's added third. And if this enzyme changes the color of the substrate to a more brighter color, so we're seeing not any clearness, we're seeing a color now, that indicates that this person has been detected for the SARS-CoV-2 antibodies, because we know that that means that the secondary antibody also binded. Okay, so a case given to me by my professor was that 7 individuals decided to break quarantine by having a barbecue, and followed by this, about a week later, they came down with some COVID symptoms, and they were tested using the RT-PCR and ELISA, as well as getting blood drawn to do an antibody titer. So because of my knowledge of knowing how easily this virus spreads, we know through the respiratory droplets, through the aerosol in the air that we can't even see, I predict that most of these, if not all, will test positive for COVID-19. So my hypothesis was that if the ELISA test demonstrates a bright color in its plate, then this particular individual will have tested positive for COVID-19. And if the RT-PCR test reflects fluorescent light in a particular individual as well, then he or she will also have tested positive for COVID-19. So these are the results of the RT-PCR testing, and we can kind of see now these fluorescent lights I've been talking about, and we can see that each of these wells are labeled by these. So the positive control, we can see that this is what we have when it's positive, and we can see that 3 of these individuals, Maura, Johnny, and Roland, all tested positive for having SARS-CoV-2 detected by this fluorescent light through their RNA gene. And we can see that 3 of them did not. David, Patrick, and Alexa didn't have these RNA samples, but doesn't necessarily mean that they don't have the virus, just means that they don't have it currently in their body. And the test to see if they did have it in their body, just not currently, we use the ELISA test. And now we can see that the little plates I've been talking about, and the brighter colors, and so we see again the positive control, what comes up when it's positive. And we can see that 4 people this time tested for the antibodies, 3 of them being the same people who tested positive in the RT-PCR test, but now we see that at least one more person have had it in the past, because we know that when these colors are bright like this, and that means that they tested positive for SARS-CoV-2 antibodies. And we see that 2 people didn't test positive, and this can either mean that they don't have the virus at all, or that their body isn't far along enough in their fight with COVID that the antibodies don't show up in their blood yet. So lastly, the antibody titers, which I showed before with the graph. The 3 separate samples they did, and the error standard deviation bars, and this is kind of to see like how many actual antibodies, like the measurements that they have, and which is pretty cool. So we can see that 2 people pretty much had a lot more than the next, 2 people had not as much as them, and 2 people barely showed any at all. So we can see here that Johnny has a little bit shown, but Alexis doesn't have any. So we know that if Johnny already tested positive for the RT-PCR, then he did have the coronavirus, but maybe his antibodies didn't show up yet because he's recently fighting it and is not far along in his fight, so the antibodies haven't showed up yet in his body. So in conclusion, individuals that currently have the SARS-CoV-2 infection were classified to be Maura, Johnny, and Roland, which we know because they came positive for the RT-PCR test. And this means that their nasopharyngeal swab contained the viral RNA of SARS-CoV-2. And we know that the individuals that had previously had SARS-CoV-2 infection but are not currently infected were David and Patrick. We know this because they both tested positive for the antibodies, and being detected by their blood for both the ELISA testing and the antibody titer. We know that both Maura and Roland tested positive for these tests too, which can mean that they're toward the end of their fight with COVID-2, since they also tested positive for RT-PCR too. And they could be towards the end if the test already detected that they have the SARS-CoV-2 in their blood. So the only individual that actually did not have SARS-CoV-2 infection yet, we know to be Alexis, and we know this because she's the only individual out of all 6 of them who tested negative for all 3 of the tests given. So that's about it, and those are my references and the F2 FDA materials, and yeah, thank you so much.